So I was given the task to introduce how we think about developing medicine for patients. And so the title of my talk is Going from the Lab to the Clinic, the process of bringing treatment to patients. So I wanted to start off with what we all dream that our process would be like. And I think this is, if it were idealistic, what would happen? We would discover something, understanding biology and be in the lab, and then in a quick, very reasonable time frame and with ease, we would be able to deliver this knowledge and these drugs to patients. But unfortunately, that simplistic process, for many good reasons, is much more convoluted. And so I think the large outline of that does stay true in the fact that we spend a lot of time doing research, trying to be thoughtful about the biology and targets and developing new ways to treat cancer. And often that's done in laboratory research. It can be done in academic centers. It can be done at pharmaceutical companies. And then once we've identified what we think maybe is a new way to fight cancer, a new molecule or a new treatment, we try to put together a lot of preclinical data to really understand if that treatment is going to be true. And that's often, as you can see here, there's little pictures of mice and petri dishes. And so we really try to understand if what we believe to be true about the biology is going to work. And we test these in different model systems. And then perhaps if it was at an academic center, we're talking with industry or pharmaceutical companies to see if somebody would be able to help us develop that drug. And if it was, in fact, developed perhaps at like a small biotech company, this is where, it can take some time to go through the chains and see if it's going to be helpful and useful before it even gets to a clinical trial. And once it gets into a clinical trial, we're, for the very first time, making a new therapy available for patients. And so, of course, our oath is to do no harm, and we first make sure that it's safe. And once we know that it's safe, then we ask, is it helpful? And so that's what those trials do. And I'm going to spend a little bit more time in a minute kind of going over that. But let's say that medicine that we've developed does turn out to be safe and helpful. That's not kind of the end. These clinical trials tell us how the medicine works and what kind of response we might see in cancer. But then it certainly goes through a lot of governmental processes, regulatory agencies, what we most commonly think of here in the United States being the FDA, before it gets to clinics and market. And even that process, after a drug is approved, sometimes can take a little bit of time, that we all get so excited when we hear, oh, a new drug is approved. But then when I go to try to get it for my patients, it may be that the factories are still being built and the medicine is coming through. So just to say that I wish it was that streamlined process, but there's a lot certainly that goes into the development of novel cancer treatments. And this slide I really liked because it's not that, unfortunately, every medicine or every great idea, ultimately comes to fruition. And, things can fall out at different points in this process, I guess is what I'll say. There are many new potential medicines, which is the point of this graph, that occur during drug discovery at the bench research, and then for various reasons, either as they're being tested in preclinical models, or perhaps even they make it to a phase 1 and maybe they're not as effective as we had hoped that they would be. And so ultimately, we really try hundreds and thousands of drugs to get that one medicine that's helpful for patients. And there's a lot more to just think about than the science. I think doctors, scientists, our pharmaceutical partners, everybody wants things to be successful to help patients. The hard part is that we're doing this, without endless resources, including finances. And the graph here on the left, and I'll note that this was a slide put together, as you can see there in the bottom right, for a presentation done at a teaching academy with Merck. And basically, they're just noting here on the left that it takes billions of dollars that have increased year after year for the development of treatment and new drug therapies in the United States. And on the right, what you don't quite see as much is that we're not exponentially increasing the number of drugs that are being approved by the FDA. If I look at this graph on the right hard, and I'm an optimist, you can maybe see that there is a trend to more or a higher number of drugs being approved over the last 10 years as compared to the 1990s. But still, in general, it is definitely costing a lot more money to develop novel therapeutics. And so to that point, this slide says, for 4% of successful projects that actually come to fruition, all that success, that 4% has to cover the rest of all the other drugs that were attempted to be developed, but for various reasons didn't make it, to FDA approval. And so they're just noting that 96% of compounds, ultimately fail. And you can see there on the right that taking like $2.6 billion to sometimes go from a new drug from concept all the way back in the lab through clinical trial, through FDA regulation, and to approval. So then, the next thing to think about, and, when we're talking about getting a new medicine, and I'm going to specifically start talking here in just a second about kidney cancer and how we think about developing medicines in kidney cancer. But this is the general timeline of how long does it take to get through that entire process. And I would say the number of years represented here is probably like if a medicine really goes quickly through the process, that this would be like a streamlined process. And so it does start here on the left all the way back at drug discovery, drug design, experiments in those tissue culture hoods in the lab, and experiments in vivo, often, some animal models that we might be testing before it reaches patients. So 8 years in the lab, if we're lucky. And then going through these various phases of clinical trials, as I mentioned, and I'll talk about in a second, looking at safety first, then efficacy, then ultimately showing hopefully in a phase 3 trial that it is helpful, and then finally getting to that regulatory stage. And so that's another at least 7 years. And again, that would be a streamlined process. So really to take a drug from discovery all the way showing safety and that it's helpful for patients, takes us 20, 25, 30 years. So this is where I wanted to highlight. I often try to explain to patients that I'm helping to take care of, if they're interested in clinical trials, what phases of clinical trials mean. So a drug has been developed in the lab, and let's say a pharmaceutical company thinks it's going to be helpful, and scientists and clinicians are also hopeful that this is going to be helpful to patients. The first thing we do is what we call a first in human or a phase 1 clinical trial. And that's where we start often we start at a low dose and test for safety. And so there, the focus is not on how does the drug work. The focus really is on making sure we think it's going to be helpful. So we're hoping that the patient benefits from it, but also really focusing on, making sure that there's no side effects, that it's tolerable. And often the most common phase 1 trial is where we'll just kind of one at a time, very slowly put patients on this new therapy, make sure there's not side effects, and try to increase the dose so that we can understand, what dosage would be safe and where we can have the most, what we call it efficacy, but the most response for that specific drug. And then if we've done that in a phase 1, it looks like it's safe, and maybe we've already seen that some patients are having benefit, then it moves on to either a phase 2 or sometimes even a phase 3. And basically what you can see here in the blue and the purple is just that each of these phases are meant to represent that we're putting more patients, we're trying this in more people to see if it will help. And ultimately, we want to get to this phase 3 trial where we're testing it in enough patients that we really feel confident that this is a medicine that will be helpful, to our patients. And then after that, it can go to FDA to be submitted for FDA approval. And there's lots of, testing and, again, making sure it's safe and, really trying to understand how is this improved over the current treatments that we have in the landscape of kidney cancer. And then ultimately, even after that, let's say the FDA approves it, often still drug companies are required to do what's called a phase 4, which is to say, okay, now outside of just these 1,000 people who it was tried on initially in the phase 3, let's see in a phase 4 trial across, broader population of individuals, how is this drug affecting people? And so continued follow-up basically is still required during this drug development phase. So now we're here to talk about kidney cancer, and I wanted to give my very broad overview of the biology and how we think about drug development in kidney cancer. So this is a busy slide, and I don't want you to worry about the specifics here, but what I like to show is that in this kind of beige area, we're going to pretend that because it's representative of that's the cancer cell. And so when I think about how do we target kidney cancer, I think sometimes we target the kidney cell, the cancer cell itself. Sometimes down in the blue and green, we target the immune cells in kidney cancer. And then sometimes we target other parts of what we call the cancer microenvironment, other things that might come in. And on the right here, we're showing a blood vessel. So there's, the way I like to explain it, there's kind of three main ways that we target kidney cancer for drugs that we've developed. There's the immune system drugs, those are checkpoint inhibitors. There's the targeted therapies that's trying to target the cancer cell, or as you can see here, all these red lines trying to target some of the recruitment of blood vessels that feed cancer cells. And then we even have a third group here on the very right, which are some newer agents that are targeting the really the underlying biology that drives kidney cancer. And I'm going to go into some more detail here in just a second about this, give you the example of the drug development of belzutifan. So this is a big timeline schema that a partner of mine, Dr. Rennie, has created. And I think it's really a nice overview to just show over the years how drug development has occurred. And as we look over this timeline, what you'll see towards the right is we're getting more and more drug approvals for the health of our patients who have, these are mostly used in metastatic or stage 4 kidney cancer. And, back in kind of before, 2004, before we had targeted therapy, which is the box of VEGF TKI era, we had cytokine immunotherapy. Then more recently, now we have checkpoint, or what this box says is new immunotherapy. And then even more recently, over the last kind of 5 or 6 years, now we're combining different treatments to help patients live longer. So just to kind of go back about that, thinking along the timeline of drug development, I mentioned that it can take kind of 20 to 30 years. And I thought this would be a really nice example for how a drug that we use in kidney cancer, how long it took for it to be discovered and then ultimately come to fruition and be approved by the FDA. So this timeline is thinking about the checkpoint inhibitor CTLA-4 that called ipilimumab, or also known as Yervoy. And this drug was originally discovered back in 1985. Not a drug, but the biology, I should say, was discovered back in 1985. And this is the first checkpoint inhibitor, so it's an immune therapy. So it took a lot of time in the laboratory to really understand, as you can see there, 1985 to 1995, understanding the role, doing tests in mice, tests in tumors before reaching the first patient there in 2000, so 15 years later. And then from 2000 to kind of 2006, what you'll see is it started then on the bottom to be developed into clinical trials and to show efficacy, or that it was helpful for patients. really first tested in patients who had melanoma, and then tested as we now have it in approval for patients with kidney cancer. So in 2010, really almost again 10 years after going into clinical trials, it had the first phase 3, meaning that large trial where lots of patients were put on this trial and received this new drug targeting the immune system, and it showed a benefit, ultimately then resulting in FDA approval in 2011 for one specific cancer. And so I thought this was just a really nice demonstration of a medicine that we know has changed the lives of so many patients with cancer, and how long it took to get that drug, or this, first the biology discovered, and then ultimately, a drug through clinical trials and to FDA approval. So because, again, this is a kidney cancer focused talk, I thought I would also really give a specific example, and one of our newest, in fact, I think it is the newest FDA approved medicine for patients who have metastatic kidney cancer, is a medicine called Welireg or belzutifan. So I'm using this as an example, and similarly trying to show you here a timeline, and I'm going back even further, all the way back to 1894, to just state that it really takes us often a long time to understand first what's going on in a patient. Why is this happening? And in this example, the biology that I was mentioning before, this HIF is really a driver of clear cell kidney cancer. A protein or gene called VHL is one of the known factors that causes tumors to form. And there are individuals who have even an inherited syndrome that puts them at risk for the development of kidney cancer. This is called VHL syndrome. And so that was initially identified back in the 1890s. And you can see here, takes us all the way until kind of 100 almost years later, when we were able to clone genes and identify kind of these specific genetic factors in order to, in kidney cancer, learn that it was the loss of VHL, which drove another protein called HIF, which stands for hypoxia inducible factor. And then another 10 or 20 years of discovery in the lab, understanding how this worked in patients, before ultimately a drug was identified in the early 2000s. The Nobel Prize was given out in 2019 for understanding the biology that came really and drives kidney cancer downstream of this HIF2 protein. And then what you can see here, I was trying to show on the right, first, this drug was approved for patients who have a genetically inherited kidney cancer syndrome. And then just last year, December 2023, in a large phase 3 trial, this drug also was shown to be helpful for patients who have what we call somatically mutated or not inherited kidney cancer, cancer that just comes up for other reasons. And so I might kind of give just a quick overview of this, because this is kind of getting into a lot of details, but essentially here on the right-hand side of the screen, this is just trying to emphasize that VHL, which is lost in kidney cancer, it drives so many different processes. And it's our ability now in the middle of the screen to target those two proteins that come together and drive a lot of cell cycle growth and how cells grow, how cells replicate, and then targeting that HIF, basically blocking that function from happening, has allowed us to shrink cancer, and has been helpful for patients. And it took not only understanding the biology, but it really took advancements in technology. So this was work done at UT Southwestern in the early 2000s, all the way kind of into the 2016, 2019, where to be able to develop a small molecule basically that could block that interaction of HIF2 and stop it from functioning. So even though we'd known about the biology, then it took a while for us to figure out or for these scientists to figure out how to block that. And then lastly, to highlight, and I'm not showing the phase 1 or the phase 2 data, but also to say, this is then the years that it took in testing for safety and testing for, how well does this work. So I thought I'd show you, this is the phase 3 clinical trial that got Welireg or belzutifan approved for patients whose kidney cancer had progressed from after having a prior treatment. And the way this treatment was studied was, again, it's a large study, so a lot of patients put on this study, and patients get to test the new medicine, or they get what we consider as a standard of care medicine. So the belzutifan is there on the top. It's an oral medication at 120 milligrams, and then half of patients were put on everolimus, which was considered the standard of care in this trial, and was already FDA approved. And then we look at scans every so often, and we collect information to understand how this medicine is affecting patients. And we draw curves, and maybe sometimes you all have seen these curves, and we physicians often call these Kaplan-Meier curves. And so I wanted, because I'm about to show you the data with a Kaplan-Meier curve, but I really liked the schematic that we show curves, but really what these curves is what is happening to real people getting these medicines that we're hoping are going to be helpful to them. And what you can see here in the schematic is that on the top, they're showing that an experimental drug is being tested compared to an old treatment or the old drug on the bottom. And that every time cancer maybe grows and maybe is showing that a drug is not working, then those patients represented by these little pictures along the line will come off the clinical trial. And so it's called that they progress, if it's a progression-free survival, or if unfortunately they've passed away, then these lines will drop. And so I liked the bottom here saying, this is where we have hope. This is where we want to see big differences in these curves, what we call the Kaplan-Meier curves. So going back to this phase 3 clinical trial that got Welireg approved, on the left shows progression-free survival. So what that means is we're watching on scans as patients are taking these medicines to see if the cancer is shrinking, if the cancer is stable, or if it's growing. And if it's showing, benefit, then we want the curve to, of course, be as high as possible for as long as possible. And the green curve here on the left is the new medicine Welireg or belzutifan. And what you can see is that unfortunately it doesn't work for everyone, but it does work for a portion of patients. And for some of these patients that it worked for a number of different months. So it was based on this clinical trial that the FDA approved this drug back in December here in the United States. And then we also will often show data just about how much on the scans do we see tumor shrinkage and what percentage of patients get tumor shrinkage. So that's what was called an objective response rate, highlighted there at the top. And you can see here with belzutifan, that rate was 22% compared to the control medicine that we were using, which was 3%. And then we also try to break it down because maybe somebody doesn't have shrinkage, but maybe still some patients have keeps their cancer from growing, and we call that a win sometimes. And so that's here is stable disease. So this drug was approved, and we were thrilled to have a new treatment option, for patients whose cancer is progressing. But ultimately, I think, these are obviously not the last points to this story. Every time a new drug comes out, we're grateful for it and we're so hopeful, but we then need to do other things. Think about, can we identify which are the patients who respond? Is there a biomarker that we can identify? Learn more about the side effects that come from these medicines. How can we help our patients? What are they experiencing? How can we make it more tolerable? And then often clinical trials have a lot of restrictions in order to go on that clinical trial. So we want to understand in every clinic for every patient, is that medicine behaving the same way, or are we seeing something different, after the drug is approved and available to everyone? And then lastly, unfortunately, these medicines don't work long-term, and the cancer cell often will figure out a way to become resistant and grow despite this medicine. So then understanding what are those mechanisms, can we maybe target those new mechanisms or make that drug better by taking it back to the bench and kind of going through the process again. So this was the most recent example of a drug approval in kidney cancer, and I hope it was a helpful way to kind of show you the process to take everything from kind of discovery about science in the lab all the way through clinical trials and hopefully for the help of our patients.